Alright, so in this video we're going to talk a little bit more about the actual structure of DNA, which all of those scientists that we talked about in the last video contributed to, and talk about the replication of DNA, or how DNA makes a copy of itself. So first, it's pretty important to understand the different components of DNA. If you remember from organic compounds, the monomer of DNA is a nucleotide. In fact, the monomer of all nucleic acids is a nucleotide. There are two types of nucleic acids, DNA and RNA. This video is going to focus specifically on DNA. So DNA, made up of those nucleotides, has three main parts. You have a phosphate group. We have in DNA a 5-carbon sugar that's known as deoxyribose. That 5-carbon sugar is also known as a pentose. And then attached to that sugar is going to be a nitrogen base. Now that nitrogen base can be either a purine or a pyrimidine. Purines are the two nitrogen bases, adenine and guanine. If you look, they are a double ring structure. If you compare that to pyrimidines, or cytosine and thymine, they are single ring structures. This uracil over here is unique to RNA, and we'll come back to that in the next video. If you look a little bit more closely at the structure of DNA, you can see these nucleotides joining together. If you notice, every purine matches up to a pyrimidine, A to T, G to C. That's how those base pairing rules always work. In between adenine and thymine, you're going to have two hydrogen bonds, which are really weak bonds. And then between guanine and cytosine, we're going to have 3 hydrogen bonds, which again are weak bonds. On the outside, you have the sugar phosphate backbone, which makes up the sides of the DNA. And in the middle, those hydrogen bonds and nitrogen bases would make up the rungs, if you were comparing it to a ladder. So we talked a lot about that structure, and now we want to focus on one of the most important processes that DNA goes through, and that's DNA replication. We mentioned this when we talked about the S phase of interphase, or the synthesis phase of interphase. So DNA replication is when DNA makes a copy of itself, and if you remember, thanks to Meselson and Stahl back in 1958, they experimentally proved that DNA replicated semi-conservatively. If you don't remember what that means, go back to the last video and watch that section. And it can be broken down into 3 main steps. You have the unwinding of the DNA molecule, you have the complementary base pairing, and then you have the joining of some different segments of DNA. So let's break down step 1 here, the unwinding. DNA replication, like all cell processes, is highly controlled by enzymes. So we're going to talk about several different enzymes involved in this. The first one is known as helicase. And helicase is, or function is to unzip the DNA molecule at certain points. So this is an example of DNA helicase, just unzipping or breaking apart that DNA molecule. We then have these types, or these proteins, called single-stranded binding proteins. It's represented by these little circles in this diagram that keep the DNA molecules from coming back together. It keeps them separated so DNA replication can occur in that one area. And then we have this other protein over here, or enzyme, known as topoisomerase. Its job is very similar to these SSBs, and that's just to make sure that the DNA molecule stays apart so replication can occur in that one segment of DNA. Step 2 is the complementary base pairing. So this is when we're going to actually start matching up bases after the DNA molecule is split in half. Remember those base pairing rules, A to T, G to C. So before this happens, we have an enzyme DNA primase, which places primers that the DNA polymerase, another enzyme, can recognize. A primer is a small section of RNA. It's responsible for telling the DNA polymerase where to start adding nucleotides, or matching up those bases. DNA polymerase is the actual enzyme that does that. So it adds nucleotides. It's also known as the synthesis enzyme. And again, matching up A to T, G to C. Purine to pyrimidine, purine to pyrimidine. DNA polymerase has several roles. One of those roles is, as it's adding nucleotides, is to also check itself. So to make sure it's actually hooking up A to T, G to C. And if it messes up, it can actually fix those mistakes. And what else is important to understand is that it can only add nucleotides from the 5' end to the 3' end. So what does that mean? Well, the 5' end of nucleic acids is the phosphate group. And the 3' end is going to be the end that has that free hydrogen, or excuse me, hydroxide group open, where another phosphate can come in and attach. So nucleotides can only be added to the 3' end or where there's that hydroxide available to attach another phosphate. This is important because it leads to the creation of leading and lagging strands, which is what we're going to talk about in the next diagram. So here we have a simple diagram of the DNA double helix over here before it's unwound. We have the 3' and the 5' end. If you guys recall, DNA is anti-parallel. So what does that mean? Well, one side is going to go from 3' to 5', and the other is going to run from 5' to 3'. Another way to think about that is that the phosphate group on one side is going to, on the other side is going to be that free hydroxide group. So they're opposite, running in opposite directions. Helicase comes in, it's going to unzip that DNA and form what's called a replication fork. And then DNA polymerase comes in and starts adding nucleotides. Remember, it can only do that from the 5' to 3' end. So the opposite of 3' is going to be 5'. So it can do that very quickly, linearly, without many problems. But when you look at this other end here, remember, it can only add from 5' to 3'. So because we need this other strand to be 3', DNA polymerase has to work backwards on this strand. This is called the lagging strand. So it has a primer, which again tells the polymerase where to start adding nucleotides, and it adds it towards the 3' end. Then it goes back again and adds it towards the 3' end, because it can only do it from 5' to 3'. And it does this in little segments. And those segments are called Okazaki fragments, named after the guy who discovered them. This, I'm going to post a video on Moodle for you guys to watch to kind of explain this if you're still struggling with that. But this is the difference between the leading strand and the lagging strand. Alright, so step 3, the joining. So DNA polymerase is going to have another job, which is going to go in to change those primers into DNA so it can actually be incorporated into the new daughter strands. And DNA ligase is going to glue those Okazaki fragments that we just talked about together. So because there are gaps in the lagging strand, ligase comes through and seals those gaps to make it one consistent strand. This is just another diagram to show you the different components working. Lastly, I want to talk about the difference between prokaryotes and eukaryotes, especially when it comes to DNA replication. So remember that prokaryotes have circular DNA, while eukaryotes have linear DNA. So prokaryotes, because of their circular DNA, they can actually replicate their entire genome at the same time in both directions. And so replication here until they meet in the middle. It's also pretty cool in that it can make proteins at the same time, which eukaryotes don't do. We'll talk about that in the next video. Eukaryotes are a little bit different in that we form things that are called replication bubbles, which are these little openings in the DNA strand. Each of these replication bubbles is going to undergo replication towards the replication forks, which are these little openings at the ends of the bubbles. And at the very end of this one linear DNA strand, all these bubbles are going to meet thanks to DNA polymerase. So eukaryotes replicate different segments of their DNA at the same time, and then it all becomes, they all meet together to separate out those daughter strands.